of this little part is to look at some of the ways that we can actually assess diversity and classify microbes. And yeah, there's a lot of really neat tests that you can do. So let me start up this PowerPoint so it puts the subtitles on. There we go. Okay, so there you can break down the way we classify microbes into based on genotype and based on phenotype. So genotype is what the nucleotides inside say. And so one typical thing that we would do would be to look at the genes encoding the ribosomal RNA, because those are how we can make really good phylogenetic trees. The genomic analysis would be looking for not just the ribosomal RNA genes, but looking for genes that encode a lot of other things. And so we could see what's the full range of potential proteins that this this bacterium can make. Yes, sorry, we've been talking about viruses a lot, haven't we? Okay, and then on the other side, there are the phenotypic classifications. And so those are going to be based on what a bacterium looks like and what it does in the world. So the genotype is about what's on the inside. Phenotype is about what effects it has on the world around it. And so there's going to be things like gram staining, which is a thing that you will have probably done in lab class. And it's a way that we can get a rough idea at least of what the structure of the membrane on the outside of the bacterium is. And do they have flagella, stuff like that? Do they have pili? All those sorts of things. Metabolic traits would be enzyme activity, and those are really fun tests to run. So essentially, tests like this work in this way. So you get a neutral agarose, which is just a sort of a regular substance which is not digestible by bacteria. And you're going to put your bacteria on that. And into this, you're going to put all of the trace metals that they would need to make a living. So little bits of iron and little bits of copper and things like that. And then you're going to put in everything that would keep bacteria alive, except you're not going to put in a food source. You're going to leave out what we call the carbon source. And you can put in a carbon source instead that is whatever you want to see if the bacteria can eat. So you could put in milk protein. That's a really fun one. And if you put the bacteria on top of that, they will actually dissolve the milk protein. And so it'll go from this cloudy, milky plate, and they'll just eat little clear patches in the plate as they destroy the milk protein. It's really cool to see. And if you see something like that, that tells you that they have enzymes capable of dissolving that particular milk protein, some kind of protease probably. As well, you can do this for biotechnology. So this is a key thing. If you have, let's say, a plastic that's building up in a landfill or something, and you want to figure out, is there a bacterium that has an enzyme that can break this down, you can take out the carbon source and put in little bits of ground up plastic instead and look for bacteria that are able to thrive and grow on that. Chances are the answer to most biotechnology problems is already somewhere on Earth and in some kind of bacterium. And it's really just a matter of bioprospecting. You sort of pan for gold and look for things that might be useful in an economic sense. So, how do we approach assessing diversity in microbial communities? So most of the diversity assessments that you'll hear about will be big things that a person could walk along and point at and, possibly even pick up in their hand. So this will be people counting animals or counting trees or kind of big plants. But microbial diversity is important as well, and it is very much connected to all those other parts. Essentially, the plants and animals can't live without the microbes, and some of the microbes can't live without the plants and animals, most likely. So, yeah, they're interrelated, and that's why microbial diversity is also very important. We're just not sure exactly what to do with the data yet, I would say. You can show differences between particular areas, but there's not like one number that would tell you if a piece of soil is healthy or something like that, whatever that would even mean. So anyway, the approaches are that you take your microbial community. So imagine we take a little lump of dirt, and there are going to be various microbes inside that dirt. We can do two things from it. One, we can culture, and the other, we can do a nucleic acid extraction. So this is trying to isolate DNA or possibly RNA, and then we can try to sequence that DNA, and so that'll be the molecular characterization down here. Now on the other side, culturing I think is quite interesting. Anything that grows in the world can probably be grown in the laboratory, but quite often they'll have specific requirements. For some bacteria, they can only work as a pair because they have a symbiosis with something else. And when you just pick up a little ball of soil or something, you will get, on average, about 10,000 different species of bacteria per gram of soil. And for the most part, we don't know exactly what makes most of those bacteria tick, and most of those bacteria probably could be grown in lab, but have not been grown in lab. So there might be 15 and 20, maybe even 100 out of your 10,000 that somebody has definitely seen before and grown in their lab. And all the rest of the ones that you find will mostly be new to science. Some may be variations on things we knew about, but you'll probably find something completely new, just reading out a little bit of soil. Because there are so many bacteria on the planet, it's pretty hard still not to find something new, which is an unusual problem in science, right? But kind of a neat problem. Okay, so these nucleic acids are used as what we call biomarkers, which is just something that you can read out, and if you know one or two genes, it tells you at least something about the organism. For example, if there are only certain genes they would use in a human for something like a paternity test, or to identify a murder scene victim, something like that. And even though there's a full genome there with all those billions of bases, it's really only a couple little parts that we would tend to look at for that. And you'll get an answer that's good enough, that's close enough. That's the idea. So, yeah. The ribosomes are made of two pieces of RNA. One folds up to make a small ribosome subunit, the other folds up to make a large ribosome subunit. And then they come together, and their job is to take messenger RNA, read it, and make a protein in response to the messenger RNA. And so one of those RNAs, the 16S ribosomal RNA, this is the one you would find in the small ribosomal subunit, and it turns out to be a very good biomarker. There are regions at the beginning of the gene, right around here, and regions at the end of the gene. Let's see, there's the end. I don't know, it'd probably be right around here, that are almost universally conserved. So they would be exactly the same in any kind of animal or plant or bacterium. And remember, bacteria are very diverse. But yeah, you can get universal primers that will amplify anything, or rather amplify the ribosomal DNA from anything. So I'm saying ribosomal DNA because there's ribosomal RNA, that's the product of the gene, and then the ribosomal gene itself is written in DNA, and so that's what you're actually reading. We don't have a good way to directly sequence RNA, and so we turn it into DNA, or we use the DNA that made the RNA if we want to read out an RNA sequence. So if you were to do what I said and look at bacteria in a little lump of soil, for example, this is what you would find by something like, this is what you would find with 16S ribosomal RNA or ribosomal DNA sequence. You would get a lot of different groups, and so these are groups that contain fairly well-known things. For example, proteobacteria are the alpha, beta, gamma, delta, proteo, and epsilon proteobacteria. But then over here, the green non-sulfur bacteria, you won't ever run into them, but these are pretty well known to environmental microbiologists. And up here, these things like OP11, TM7, SE1, these are all branches of the phylogenetic tree. And they're branches where we haven't even given a proper name to the branch yet. Most or all of the members of these branches are things that have not yet been cultured. But they're out there. They're all over the place. And this is at the level of phylum. So this is, one step below kingdom. So these are really broad differences between different kinds of bacteria. And this is a typical sample. This actually looks like a fairly small sample. If they took a little bit bigger sample or broke it down by individual species, you would expect to see many thousand different kinds of bacteria. All right. And so it's used to answer questions like this. For example, so are some of these bacteria endemic or unique to a given location or soil? Yeah, there are different kinds of soil with different amounts of water and sand and looseness and clay and all sorts of stuff. And yeah, there may be bacteria that are unique to that. So a lot of what we think we know about farming and the way that plants behave is actually coming through or being modified by the bacteria in the fields. They have a very big role on what it is that's available to plants and on the health of the plants in the field. So there's some bacteria that are very important for plant growth, and there are others that are, cause diseases in plants. And so, yeah, bacteria do a lot of different stuff. And so you can answer questions like this with this sort of genotype analysis by looking at the 16S ribosomal DNA. Okay, there's another thing that you can do, and that is a technique called a rep-PCR. So rep sequences are short sequences that occur at different locations throughout the bacterial genome. And if you have the same, it's short for repeat, yeah, fantastic, or repetitive element. If you have a sequence that you know and a sequence that's repeated throughout the genome, then all you have to do is put a primer over each one of those sequences and you will amplify something. You will amplify little bits of DNA. And once you've amplified it, then you can read it out. So by amplify, I mean PCR technique. But the nice thing with this rep PCR is that you don't actually have to know what it is you're looking at, and you can look at an entire community at one time. Develop a thing called a genomic footprint. So let's zoom into this. This is a rep PCR gel, and this is a way to look at genetics of a lot of different genes at once. And so we've got a couple different species here, and so species 1 looks like it's usually the same. Species 2, look at that, is quite different. And so if you had a mix of species 1 and species 2, you'd be able to tell the difference between that and a pure clone of 1 or 2. And if you had a mix of a lot of different species, you're actually able to read out the entire population. And you can get kind of a fingerprint for a particular sample. Now, you won't know the actual sequence of the DNA in that sample, but once you have the this rep PCR gel, it'll actually tell you, you can do statistical analysis on it and see whether or not one location is about the same as another one or is really different from another one, just whether they're significantly different or similar or neither. And that can help. That can help us understand the world. And so from this, we've figured out that different populations of bacteria called Pseudomonas can actually colonize the root area, that's the rhizosphere, and the same plants grown in different soils. So these bacteria called Pseudomonas are, and relatives of Pseudomonas, like Burkholderia and Ralstonia, for example, they live around plant roots normally and occasionally cause plant diseases, but also potentially, just use the plant in a sort of a commensal kind of way where they don't really hurt the plant too much. And this shows that in certain types of soil, you'll actually get slightly different versions of these bacteria, of these pseudomonads sitting around the plant roots. So that's interesting. Okay, that's the end of that little part. And thank you very much.